Alright, now we're going to talk about chromosomal mutations. And the first one we're going to talk about is the most common, and that is non-disjunction. Non-disjunction can occur in either meiosis 1 or meiosis 2. We're going to look at meiosis 1 first. In our diagram here, we can see that non-disjunction occurs when a pair of homologous chromosomes fails to separate. In this cell here, we see that these homologous chromosomes did not separate. This one right here should be over here, like we see in a normal meiosis 1. That didn't happen, so that's a non-disjunction there in meiosis 1. What that makes happen in meiosis 2, when we have our new daughter cells created, is that this daughter cell is missing some chromosomes, whereas this one over here has an extra copy. Now, after that goes through normal meiosis, we don't see any other non-disjunction occurring here. We end up getting two daughter cells from this parent cell, and we have an extra chromosome here. And we have an extra chromosome here as well. It should have one short, one long. And over here, we're missing a chromosome. So that's what that N-1 means. We're missing one chromosome. The N+1 means we have one extra chromosome there. These will all be abnormal haploid cells. And these haploid cells will be gametes, so we have abnormal gametes. So if these sex cells were going to be used during conception, the possibility of the child either growing up and having some type of abnormality, some disorder, would be possible. Also, a chance of miscarriage as well. If we look over in normal meiosis 1 over here, and we have non-disjunction occurring in meiosis 2, what ends up happening is we have a pair of sister chromatids that fail to separate. So before is this homologous chromosomes. Now we have our chromatids that are failing to separate. So this other chromatid should go over here, as we see in a normal meiosis that occurs here. But in this cell, non-disjunction occurred. What that leads to, as we see, is one gamete will receive two of the same type of chromosomes, right there. We'll have one gamete that will not receive a copy of a chromosome, right there. We'll have two other gametes that will be normal. Now again, I said this happens in 10 to 30% of human conceptions, and that is the main cause of miscarriage as well. Now, we're going to take a particular look at the 21st chromosome. So we have 23 pairs. So what happens in that 21st pair when we have non-disjunction? Well, the 21st chromosome, when it's not evenly distributed, we have one gamete that will have two copies of the 21st chromosome. The person will actually have 47 total chromosomes then, if that gamete is used during conception. So normally we have 46, but this person will have 47. We call this trisomy 21, tri, 3, and then we have 21 denoting our 21st chromosome. This is also known as Down syndrome, and it's actually the most common chromosome abnormality. Again, this is a type of non-disjunction. It occurs in 1 out of 850 births. And as we see here, that infants with Down syndrome, the occurrence increases sharply after the age of 35, especially after 40. Now here's a trisomy 21 karyotype. A karyotype is a visual representation of all of the chromosomes. This can be done in the lab, and we can use equipment to take a picture like this. And we can then use that and we can analyze the chromosomes and their structures. And what we see here in the 21st chromosome pair down here is we have an extra one. Now, these 1 through 22 pairs, these are known as autosomes. And this one all by itself over here, this pair by itself, this is known as our sex chromosomes. So we have a lot more autosomes than we have sex chromosomes. And this person here have two X chromosomes, so they would be a female. If we had an X and a Y is much smaller, then it would be a male. And actually this is going to lead us into our next discussion, and that's non-disjunction of sex chromosomes. So normally you're XX or XY, female XX or male XY. There is a possibility of a male to have two X chromosomes due to that non-disjunction. That's called Klinefelter syndrome. And a male that has this syndrome has small testes, subnormal intelligence, and female body characteristics such as enlarged breast. We can also see this as XXYY, XXXY, and XXXXY as well. XYY. What if you have an extra Y chromosome? Well, that's no defined syndrome, but we do notice that males that have that are taller than usual. There's a possibility for a female to have an extra X chromosome. Now, they cannot be distinguished physically, but genetically we can see that during karyotype. And it is also possible for a female to have lost a chromosome due to that non-disjunction, that extra, the other X chromosome went into another gamete, but the gamete that was used was missing a chromosome, the sex chromosome. And so what we have here is a person that was born, a female, that just has one X. And over here, we can also denote that as XO, O meaning that you're missing the other chromosome. Then we call this Turner syndrome. And females with Turner syndrome are shorter, sterile, they have a web of skin between the neck and shoulders, and they also have normal intelligence as well. Now this is when someone that has 45 chromosomes and it's not fatal. Usually if you're missing a chromosome, that's a fatal effect. But here we have 45 chromosomes and not fatal. What that tells us is that the Y chromosome, it really isn't as important for development. And when you have two X chromosomes, you can actually survive with just one, because what happens when you have two is that there are sites on one of the chromosomes that become inactivated. But we'll discuss that stuff when we get into genetics next unit. Now, that's when you have, what we just talked about is when you have chromosomes that the whole entire chromosome goes to a cell that it shouldn't go to, and that leaves some with extra chromosomes or some with fewer chromosomes. Now we're going to take a look at what about the chromosome itself. If something on the chromosome gets changed, and not just where the chromosome goes. Well, we have deletion here, and it's exactly what it sounds like. A segment of the chromosome is removed, is deleted, and we can see that in our picture here. This section here was deleted, and now it's much shorter, so we're missing a whole lot of information. Duplication is exactly what it sounds like. This section here is duplicated, and now we have an extra occurrence of that information that's found in that segment. We have inversion, and inversion is when you just take two segments and they are flipped, as we see right there, and reinserted into its original orientation. And we have reciprocal translocation, and that's when you have two non-homologous chromosomes, right? So they're not like each other. And we see that here, the red and the blue, and also the shorter length and the longer length. And we're going to take this whole section here and this whole section here, and they're going to switch. And that's reciprocal translocation there. All right. Well, that's take a look at some of our chromosomal mutations. If you have any questions, let me know in class or let me know in the comment section. Thanks.